with my La Follette School of Public Affairs hat on, like to acknowledge Herb Cole, who's made substantial contributions to our school with the focus on bringing the work we do to the broader Wisconsin population. So I've been doing this type of talk around the state, and I'm really happy to share some of my thoughts with you and have some conversation. So a lot has happened in genetics in the last 20 years or so. This figure, you can barely see the actual human chromosomes lined up on the figure. What the main point of it is every little dot here is a new finding from genetics of a particular area in the genome that's linked to something important like cardiovascular disease or Alzheimer's disease or height or weight or so many things. So as in total, it's the number of findings is approaching, I think, 400,000, populated across over 6,000 publications in the last 15 years. So this is, exhibit A of a revolution happening in genetics and what genetics might be teaching us about our own important outcomes that we care about. And so way back five years ago now, I published a book with a sociologist, Dalton Conley, to summarize what we thought we knew then. This was in 2017. And the reason I highlight this is because you can see in the end of the subtitle, it says the future, and we speculated about what's going to happen in this far-reaching future. And we were way off, which is to say that everything that we were speculating on is like now in our rearview window. And this is just so fast-moving, the science. And what I want to stress is both the innovation, the excitement that we should all have, but also the policy questions that it raises that we're probably 20 years behind in actually thinking about. So I'll be going back and forth between those two ideas here. I'm going to first set up the claim of a revolution and a couple of the components that I think are behind this revolution in the last 20 years by first reminding you of, I think, about three facts that are going to be important in thinking about policy implications. So the first is essentially like is the size of the genome that we're talking about. 3 billion letter, I'm going to call them letters A, C's, and T's, and G's across the genome, 3 billion pairs. There's a lot of information packed in each one of our cells. And the revolution here is to try to get an understanding of all these 3 billion letters. So that's setting up the scope of the information that's possible. It turns out that nearly all of the letters, but not all, so both those things are important, are the same in each and every one of us, all humans. I think that's a nice starting point to say how much we have in common. I think it's also true that around 99% of all our letters are the same with chimpanzees, so which is to say that some of these letter differences are really important. One important letter difference, this is just one of those 3 billion letters. If I read off letters, I said there's C's and T's and G's and A's. This is if I zoom in on a very narrow part of those 3 billion letters, I can observe C's and A's and T's and G's in normal cells. And then I hop down here, and all these are the same except for this one letter. And that letter is really consequential here because it determines if you have a sickle cell or a normal cell. Another similar small difference across our genomes will decide whether you are lactose intolerant or not. So some of these letters really matter, I guess is the point. And the other point for our policy and for what we're doing with these letters and this information is that these letters are permanent. You'll notice that unlike the other statements, there's a little asterisk here because we might all recognize that permanent in the era of gene editing, there's an open question here, which I won't talk about very much today, but just to highlight that I think we're way behind in thinking about what we're gaining from the genetic revolution. We're even farther behind probably in thinking about the component of that revolution that's about gene editing among humans. So I'll put that aside now just to say that I understand there's an asterisk there. You should understand that there's an asterisk there about the permanence of these letters over your life. So I'm going to transition in the next few slides about claiming that there's a genomic revolution that is becoming or has implications for a social genomics revolution, which is to say there's many social implications, policy implications we need to be considering. Okay, so I gave you the genetic facts, only a couple of them, but I think those are the most important for today. And then I'm going to talk a little bit about what's the source of revolution. So the genetic facts were true forever. Nothing new from that last slide except for the potential editing. But what's new is technology around understanding those letters. And then what's also new is working with those letters using quote-unquote big data techniques to examine statistical models that are very complicated now. But I'm going to focus on the new policy questions that these facts plus technology plus statistics seem to be pointing to. What are the improvements? I think a slide on this. So you're all familiar with what I'm about to say, but let me set the stage. That in the last 20 years, prices change. That's the new information here. But in particular, I'm just reminding you that some prices go down, like televisions are cheaper for the quality that you get now than 20 years ago by a lot. This graph is suggesting that like the real price of a television for the quality that you get has dropped by about 90% in the last 2 decades. You also won't be surprised to see on the top of this figure, college costs are among those that are rising a lot over the last 20 years. Child care, medical care. So prices change, especially you hear about inflation every day now, but even before inflation was the top news item, this has been true for forever, but in particular the last 20 years, there's been a lot of changes in price. Now this seems, I'm going to call this normal change in price. These are things that we have lived through in the last 20, 25 years, and you've lived through before that, other prices that have changed a lot. We have never lived through the price changes that have happened on the technology around sequencing and gathering information from DNA. So Moore's law, which is the fastest thing we've ever seen in terms of price per unit change, which is computing capacity, which doubles every year or less, which is to say it's been cheaper very quickly to get better and better computers. So that's a very fast phenomenon in the last several decades. But the lines under that are the price of sequencing human DNA or other human DNA for today, which has fallen by a factor of 100 million times in the last 2 decades. So we think about TVs, being cheaper. This has gone from incredibly expensive to sequence one human to like $100. And that has a lot of, you think something boring like price change. In the other room about the local branch of all of us, which you see as a little pointer of a few years ago the goal was 1 million people. I have a little note here that Ancestry DNA during the weekend of Thanksgiving 2017 had 1.5 million genetic tests run because they had a sale. Massive data, right? Massive data collection. I'll more quickly go through the statistical improvements, which is just to say, with all this big data, there's some way you have to wrangle it to make sense of it. And there's been a lot of work, a lot here, but all over the world about doing that, about wrangling these enormous data sets to try to make sense and to make predictions based on DNA. And that's done, so this is our friend from the first slide here, our human chromosomes, and how it's done is statistical analysis, point by point. You just run through the whole genome, location by location, and ask if you have an A there versus a T, a C there versus a G, are you taller or shorter? Are you more likely to have Alzheimer's or not? Are you higher weight, lower weight? So on, you have blue eyes, non-blue eyes, so on and so forth, all the way through, over and over again. And this is where you get tens of thousands or millions of analysis that then you have to make sense of. This is a schematic of lots of analysis and then making sense of that through prediction. So I'll have a couple examples of that: scanning, looking, and then using that information for prediction. So here's a scanning, looking, where the chromosomes are colored. And like I said, every location, yes, no, does this person have this condition? Yes, no, yes, no. And this is an interesting example where there's like nothing across the whole genome except one location or one little cluster of locations. And this would be a simple case. This is the case of whether this is the self-report of your ability to detect a strange odor from urine after asparagus consumption. So it's a simple genetic architecture here. This is, if you looked across your whole genome, nothing matters for that outcome except right where you see this huge peak. Okay. So this is a simple case. Most things are very simple. A little bit less simple are varieties of allergies. So upper left is egg, upper right is cow's milk. Upper left is any food allergy, upper right is peanut allergy, lower left is egg allergy, lower right is milk. What you see is still some simplicity, which is you see some big peaks and a lot of background noise, but you can see some differences across these figures, and more peaks, which is to say more complexity, more polygenicity, more many genes mattering. And this is going to be more similar to everything we care about than the asparagus example. This is actually going to be still too simple. Most of everything we care about is even more complicated than these figures. Here's a figure of just how many of these figures are being produced. So it's more than one per day. There's some scientific team every day that's releasing results about a new outcome. It could be asparagus, it could be allergies, but some other outcome could be blue eyes that has this in the background, a scan across the genome, where in the genome. Doesn't matter whether you have an A versus a T, a C versus a G. So this is 3,000 such analyses, and you can see a huge increase in the flattening with a lot every day happening. Okay, now why does this matter? This is like in the background, the scientists are busy like pushing buttons on their computer and swabbing cheeks and genotyping the saliva samples. One reason is the use in the clinic, which the cartoon on the lower left here is such a use. If you've been genotyped, you could, and many of you have experienced something like this, where you sat down with your physician and the physician says, oh, looks like based on your genetics, you're at higher risk for X, for cardiovascular disease, or for Alzheimer's, or anything. So in the background here, the reason is that it's possible is it's so cheap to collect and analyze your DNA, and there's so many studies now in the background that tells the physicians what to do with all those A's and C's and T's and G's that they've sampled from your saliva, and then could make clinical decisions based on this type of information. And this is definitely in the field for many types of cancer, especially breast cancer, where there's both big hits, BRCA1, BRCA2, but also all across the genome, small effects that really add up and matter for Alzheimer's disease, like I said, breast cancer, and so on. So this is still usually at this point in the talk, people are still pretty comfortable in nodding. Okay, like, yep, this is really exciting, great news. Now I'm going to start doing a little bit of shift of maybe some unanticipated consequences. And you've already probably are going to know where I'm going, which is to say you already know that 1 in 20 Americans have been genotyped by a private company. So this isn't your doctor. And I just did a search yesterday about like answering the question, what could you do with your So this is now, what could you do with your own genetic information, which is to say, this is very much a consumer-directed market where, at home, you spit in a cup, you send it out, some company genotypes it, they'll send it back, and then the question is this going to be helpful for my, for what types of drugs or what types of prescriptions I do? This is with no interaction with the medical system or the clinic, right? You can spit in a cup, send it out, they'll send back all your A's and C's and T's and G's. A company like this will take those A's and C's and T's and G's and say, oh, we predict you should have this medication, not that medication. We predict your metabolism is such that the medication that you're on is not the right answer, and so on and so forth. So this is a slight difference than this, where you're talking with your clinician. And again, in the backbone of this difference of now being completely outside of the medical setting is the background being it's really easy to collect your information, your DNA, sequence it or genotype it, send it back to you, and make these predictions. So I'll just now transition into this fact that there's nothing special about cardiovascular disease in everything I've said. There's nothing special about Alzheimer's disease. In fact, we can do all the work I just described on any outcome you can imagine, and are, and have been for many years that you don't know about. So the same steps used to analyze and predict these procedures in the clinic can be done with no interactions with the doctors, as I've already said, but with outcomes that have not very much to do with health. And here's where I think the policy questions really come in about what kind of protections we would like our government enacting for us to use our own information or have others use our information in discriminating for or against us. Right? So if you're in the clinic and the doctor says do A versus B based on your genotype, that's discriminating, and we probably like that kind of discrimination. But it's also true that your life insurance company can discriminate against you in providing either any policy at all or the price of policies, and there's no government regulation around that. So if your life insurance company asks you to be genotyped and they find out you have high risk of Alzheimer's, they can and will use that information for you or against you. There's no regulation. And now, one more step. So this is the plot like the asparagus plot, but this is predicting how far in school you go. This was done over 6 years ago. So this is only 300,000 people in the background and you can see so many peaks. There are, and this was published in Nature, one of the leading science magazines. This is across the genome. You can see the headline. There's 74 locations that really matter for schooling. So this is ancient history. This was 2016. This summer, 3 million people are underneath this educational attainment genome-wide association study. There's 4,000 different locations in the genome that have these huge peaks. And the same things that can be done with cardiovascular disease inside or outside the clinic can be done in predicting your schooling inside or outside the clinic. And by outside the clinic, I mean you can get your genotype, you can get your information, your DNA genotype by 23andMe or some other service. They can send it back to you. You can go to another company and they can tell you what your genetic risk for going to higher education is. And similarly to like who do we want to use the information on our Alzheimer's risk for life insurance, who do we want to use the information about how far in school we're going to go predicted by our genetics? You won't be surprised, I think, that there's no regulation on this. So do we want precision medicine, which I think a survey would suggest many people are enthusiastic about, to become precision policy without us having much say in it. Right now we don't have much say, or we haven't had much say. So some examples, the only areas right now that are off limits are our health insurer and our employer by a law from 2003 called GINA from under George W. Bush's presidency, is Genetic Information Non-Discrimination Act, is that there's 2 areas in your life that you're protected. your health insurer, it would be illegal for your health insurers, not that they cannot do it, but it is illegal for them to discriminate against you based on your genotype. It's also illegal for your employer to discriminate. And that's it. That's the end of that sentence of who is illegal to discriminate against you based on your genotype. And the reason I had this here is the article from 2017 was describing that this GINA Act was one vote away from being totally dismantled. This is actually John McCain was, while he was living, was one of the decisive votes on keeping GINA, because health insurers would prefer not to have their hands tied. Employers would prefer to not have their hands tied by the government in using your genetic information for you or against you. A really important book in academic circles from one of the founders of behavioral genetics, Robert Plomin in the UK, most of this book is the case for precision education. Essentially taking, genotyping children. You'll recall that your letters are the same when you were born to now. So you can certainly genotype children and make the same predictions of years of schooling as I showed earlier, but then make education decisions for children based on their genotype. Again, this is something that could have been done almost 10 years ago. We're way past the conversation, so I think it's really urgent to think about this question. So this would be a book to go for, the case to be made for. This would be the early screening for dyslexia. You'll recognize that children, very early on, before they can talk, they can still be genotyped. And you could potentially intervene earlier than you could if you were just based your clinical or screening decisions on language that you actually hear and see. You can do this, at day one. Where's another area that we should think about, and this is going back to the first couple slides about these being permanent letters, and what else might we want to predict based on all these letters? And the answer is during pregnancy. So these are a very small subset of all the predictions one could make about fetuses, because it's the same DNA when you were a fetus versus now. It's the same, my prediction for you for your years of schooling is the same now versus when you're 20, versus when you're 5, versus when you were 0, versus when you were in the womb. And the technology is such that very early on, I have a little schematic here about day 3, 4, 5, where there can be a relatively non-invasive procedure, relatively is underlined, where you just need one cell from the growing fetus to do all the genotyping I just described. And you won't be surprised when I answer, what laws are for or against this? 0. And there's many companies. Right now, the companies that I know about, they're all over the world, but one that comes to mind is in California. The services they're offering now are different than what they could offer, which is say they could offer everything, education, IQ, height, everything. Right now, they're offering sex and eye color for $200. So turn to the future. It's more of this, right? The data is getting better, the predictions are going to get better. There will be more and more scope of what areas in our lives have the opportunity, because they have the background data, to make these predictions, and therefore at least have the possibility of treating people differently based on their genetics. So the scope is getting wider. It's not just education. It's certainly criminal justice. It's health. It's really, it's really not limited. I can't think of many where in your life there would be a hard line here. So I think this is why these conversations are really important. And again, with my La Follette School of Public Affairs hat on, this is what we partly talk about on the policy side around privacy. Who gets it? Who do you share with? Here's a list of possibilities. I already told you employers and health insurers have been decided temporarily. Others have not, right? Life insurers are explicitly able to discriminate. who has access to these technologies on the front end, about who gets genotyped, and where in our lives so related to privacy, something you should always also have in your mind is breaches of data security. So here's one that you'd be familiar with. You know, big company has a data breach, and social security numbers and addresses and billing statements are released, right? And then you get a notice to say, you know, you should be careful. So the thing I'd say is different about your DNA being breached is that unlike your address or your billing history or your name or your social security number even, your DNA is permanent. You could move, you could change your name, you can change your behaviors in terms of like your credit billing history, and so you're not changing your DNA. If your Alzheimer's disease risk gets out, it's out. It's never taken back. And excuse me, this is one of many papers that suggests that the data are getting so big that you are already in the data, which means that if your brother is in the data, you're in the data. If your cousins are in the data, you're in the data because of the genetic relatedness of your cousins and brothers and family. When they make their decision to get into 23andMe, they're partially making a decision for you. And that's just a really challenging policy question, right? So privacy, access, we could spend the whole day on all the policy questions. What I'm going to leave you with is a little bit more, I think, good feeling, which is that UW is like on top of this. So many of my colleagues have a social genomics initiative here where we're hiring faculty in this area. I've listed some in their disciplines, which is to say it's a wide spread of both people who do statistical genetics, who do sociology, who do public policy, who do economics. We have 30 students across many programs that are all working on these and have courses in the La Follette School of Public Affairs on genetic privacy. So we're very much working hard, but I'll wrap up to say we just appreciate your input and your questions and comments in feeding back to our work. So I'll thank my funders, thank again Carol, and open it up for questions.